speaker will be Cristina Viriato, Unesp, Brazil. Yes.
Okay, can you see my screen?
Yeah, can you just click on F5? Yeah, slides mode, presentation mode. Yes, just go ahead.
Okay, thank you.
Thank you.
Good afternoon. My name is Cristina Viriato. I'm a PhD student at São Paulo State University. And I'm going to present you the main results of my master thesis, which generated an article published in Chemosphere Journal entitled Evaluation of the Potential Teratogenic and Toxic Effect of the Herbicide Sulfur-D GMA 806 in bullfrog embryos and tadpoles, Lithobates catesbeianus. So the growing demand for food in the world has stimulated agricultural activity, which often requires the use of pesticides. And these pesticides can be dangerous to human health and to the environment, causing ecological problems, and can contaminate water and soil and produce toxic effects on organisms. And one of the most used pesticides in the world are the herbicides. And between herbicides, one of the most used is 2,4-D, 2,4-dichlorophenolxacetic acid. It is the most one of the most used because of its low cost and good performance. And this herbicide is the most used for the control of broadleaf weeds in several cultivations as soybean, rice, corn, and sugarcane. And it is classified as extremely toxic to humans because it is an eye irritant. And in 2018, International Agency for Research on Cancer classified this herbicide as possibly carcinogenic to humans. Why possibly? Because there is inadequate evidences in humans and limited evidence in experimental animals. So here in Brazil, it is classified as dangerous to the environment, and it is slightly toxic to fish and aquatic invertebrates. This herbicide half-life in water can exceed 120 days in anaerobic conditions, but it can be degraded in a few days if found in the surface waters. This herbicide is found in surface waters in several countries in the world and also in drinking water supply systems. Here in Brazil, in South Brazil, there is a study that quantified 79 pesticides in the main course of Guaporé River in Rio Grande do Sul state. And the most found pesticide among these 79 pesticides was 2,4-D and had the highest concentration, almost 0.1 milligram per liter. And it was in areas of intensive production of soybean and winter cereals, so demonstrating a high ecological risk. Toxicological studies have been carried out using 2,4-D in several organisms, such as rats, demonstrating histopathological alterations, and fish, demonstrating oxidative stress, and in amphibians, demonstrating decreases in activity, growth, and developmental rates. In amphibian larvae, studies also showed mutagenic effects, harmful changes to biochemical, physiological, behavioral aspects, and morphological abnormalities. Amphibians are frequently used as a biological indicator to the environmental health status due to their life cycle, both in water and terrestrial habitats. And recent studies are showing that pesticides are one of the causes to the decline of amphibians. And studies are showing also the importance of toxicological tests mainly in reproduction and early developments in these animals. So this present study followed the 3 R's principle: replacement, reduction, and refinement. Replacement, which aims to replace adult animals with larvae, initial phases of life, larvae and embryos. Reduction, which aims to reduce the number of the animals tested in the experiments. And refinement, which aims eliminate the pain and suffering of these animals through anesthetics and analgesics. So the objective of this work was to evaluate the potential teratogenic and toxic effect of the herbicide 2,4-D DMA 806 in bullfrog embryos and tadpoles. So this study was approved by Ethics Committee on Animal Experimentation of the Fisheries Institute, São Paulo, Brazil. And the substance tested is the commercial formula of 2,4-D acid in dimethylamine salt form in 806 grams per liter. And the animals used in these experiments were embryos and tadpoles of Lithobates catesbeianus, bullfrog, and they were obtained at experimental frog farm in the city of Pindamonhangaba, Brazil. So for the embryos, we used FETAX test, frog embryo teratogenesis assay, Xenopus. This test was created to Xenopus levis, this species of frog, and the test exposed the embryos since blastula stage to tadpole stage in petri dishes. 25 eggs we select for each petri dishes, and this FETAX test was adapted to Lithobates catesbeianus. And with this test, we can find LC50 median lethal concentration, EC50 median effective concentration, TI teratogenic index, which means LC50 divided by EC50, and also we can find minimum concentration to inhibit growth by measuring the head-tail total length tadpole. So our test we used for replicates for negative control just with FETAX solution. Negative control 2 only with cultivation water from the farm, 2 replicates. Positive control, we use cyclophosphamide so we can compare, so we can see the malformations of the embryos, and range from 150 to 1,200 milligrams of 2,4-D DMA per liter in FETAX solution. Here are the examples of our petri dishes, and so here we can find teratogenic index. For the tadpoles, we performed acute and chronic toxicity tests in stages 31 to 36 of the tables Gosner's table, sorry. And to perform this test, we followed the American Society for Testing Materials, and for the physical and chemical parameters of the water, we followed standard methods for the examination of water and wastewater. The acute test lasted 96 hours in a static system with 4 replicates and treatments ranging from 0 to 950 milligrams of 2,4-D DMA per liter. And so we can determine the LC50. After determining LC50, we started the chronic test, which lasted 49 days. In a semi-static system, the solution was renewed once a week, and we use 4 replicates for each treatment that range. The 4 treatments were 0 milligrams per liter, LC50 divided by 2, LC50 divided by 10, and LC50 divided by 100. We choose this concentration because this special formula concentration we don't have here in Brazilian waters, the monitoring. And the tadpoles in chronic tests were fed with ration probably once a day, every day. At the end of the test, 12 animals from each treatment were randomly collected for blood collection. After applying lidocaine, they were anesthetized with eugenol on ice and euthanized for organ collection. The organs were liver, spleen, gills, kidney, and skin. So for the chronic test, we evaluated healthiness, hematological profile, histopathological analysis. For the healthiness, we evaluated hepatosomatic and splenosomatic indexes, which means the liver weight divided by animal weight and spleen weight divided by animal weight, following Quentel and Olbach methodology. And hematological profile, we determined erythrocyte count, hemoglobin rate, mean corpuscular hemoglobin, which means hemoglobin rate divided by erythrocyte count. The total leukocyte count and differential leukocyte count were also determined, following Hilbrecht and Smith methodology. And we also performed the histopathological standard analysis with gills, skin, and kidney of the tadpoles. So statistical analysis, we use a trimix spermocarbon for LC50 and EC50, student C test for minimal concentration to inhibit growth, and 1 ANOVA to the other parameters in chronic tests. So our results to FETAX test were a high concentration of 2,4-D DMA. Indicated that this herbicide is minimally toxic according to G8S global harmonized system. And also this herbicide commercial formula has a low teratogenicity according to ASTM. But we could find some alterations, deformation of the tail and abdomen of the tadpoles in concentration of 600 milligrams per liter, 70, 50, and 750 and 900 milligrams per liter. And it can be due to an interruption of a specific signaling pathway for visceral morphogenesis. So the result of minimum concentration to inhibit growth was 150 milligrams per liter, and which was the lowest concentration tested in this study. And we can observe a gradual decrease of the total length of the embryos. And more studies have to be done about the total length, about the minimum concentration to inhibit growth, because it could be lower than this tested in this study. The actual test for LC50 was 700 milligrams per liter. It is a high value, so it is also a herbicide minimally toxic to tadpoles. And we performed the chronic test with 4 treatments: control without product addition, 7 milligrams per liter, 70 milligrams per liter, and also 350 milligrams per liter. In the end of the test, the chronic test, there was not tadpole mortality, and the animals were at stage 38 to 40, according to Gosner's table. The healthiness, there was no significant difference between treatments regarding to hepatosomatic index, but there was a difference regarding to splenosomatic index. We can see here in the graphic. There was a decrease about regarding to the weight, the spleen weight of the animals in relation to the control. And a study that tested rats with 2,4-D demonstrated the same result as our study, and showing the decrease of the spleen weight. To the hematological profile, there was an increase only in 350 treatment about leukocytes, total leukocyte count, and lymphocytes and neutrophils. And I studied that exposed rat to 4-D by via dermal route concluded that this herbicide is a respiratory allergen, and they had and they showed the same increase in leukocytes, lymphocytes, and neutrophils. The number of erythrocytes and hemoglobin rate showed an increase to the treatments of 2,4-D, and these both increases could be polycythemia, which can be related to the decrease of the spleen weight, demonstrating physiological stress by the demand of oxygen. So in histopathological analysis, were found alterations in the gills of the animals. So here in figure A, they are comparative photomicrograph of gill tufts of bullfrogs. So in the figure A, we have the figure A represents negative control, secondary gill tuft with ramifications, Figure B represents the treatment 7 milligrams per liter, and we can see widening of intercellular spaces. And here, widening intercellular spaces with the appearance of a large vacuum in the treatment 70 milligrams per liter. And in figure G represents the treatment 350 milligrams per liter, and we can see hyperplasia and fusions of the tufts. In kidney, there are some alterations. Here, there are a comparative photomicrograph of kidneys of bullfrog tadpoles exposed for 49 days to the herbicide 2,4-D DMA. So here we have the negative control. You can see here the renal tubes, hematopoietic cells here, and the Bowman's capsule. So figure B, figure B represents 7 milligrams per liter of 2,4-D. And we can see tissue disorganization and lymphocytic infiltration here. In figure C, we can represent the 70 milligrams per liter treatment. And here we can see the hypertrophy of renal tubules and cell with no loss of nucleus. And here in figure D, we can see intense tissue disorganization with loss of cellular structures. Also in skin, we found alterations. Here is the control group, figure A. We can see the basal cells, skin cells, apical cells, the glands here, and connective tissues 1 and 2. Here in figure B represents 7 milligrams per liter treatment, and we can see epithelial hyperplasia here. And in figure C represents 70 milligrams per liter treatment. And here we can see the dermal cells with pigments. And in figure D, we can see the intense pigmentation and also the destruction of epithelial cells layer. And we can see the connective tissue too here because of the destruction of the epithelial layer. So we concluded that this herbicide commercial formula is not acutely toxic to embryos and tadpoles of Lithobates catesbeianus, but it induces to inhibition of embryonic growth. And in chronic tests, physiological stress and dehydration, which may be associated with the hematological profile and gill tufts widening intercellular space and consequent tuft fusions, characterizing this pesticide as a respiratory allergen for Lithobates catesbeianus tadpoles. So considering that amphibians are bioindicator animals of environmental quality and recognized sentinel animals of the aquatic environment, we recommend precautionary measures for prolonged exposure of organisms to this chemical agent. Here is my group. Thank you. Here is my group. Fernanda Menezes França and Claudia Mares, my advisors, my colleague Diego Salles, and researcher Adriana Sassioto. Thank you.
Yes, thank you, Cristina. So, thanks for your time and presentation. Is there any questions from audience? Yes, Christina.